Assalamu alaikum. How are you all? Welcome back to our online classes. Hope our lessons are clear to all of you. First lesson you have written here, isn't it? You have one. Second lesson, third lesson. I'm giving you thorough explanation. If you listen to it and do the homeworks on time properly, You'll be able to answer any question. Read the lessons. Again, I'm telling all the lessons thoroughly, properly. Any question can come from anywhere. Okay. Lesson animal tissue. Got it? We'll continue with it. What we were doing. So, children, we were studying about blood platelets. 5 ml of blood is necessary. Why is blood red in color? Because of hemoglobin. Clear? I have given that question answers also. Homework also, if you do properly, read the lessons properly, listen to the explanation. Any question you can answer easily. Okay. The foreign materials which are destroyed inside the cells are called scavengers. Blood platelets are a separate group of cells which do not have a nucleus. They are disc-like projections. Whenever a blood vessel is injured, platelets accumulate at the site of injury and help in the formation of a blood clot. The clot seals the wounds in blood vessels and prevents further blood loss. Your sisters, brothers are not your relatives. This will shock you, but it is true. They don't have same blood group. Everyone have different blood group. Landsteiner, a German doctor, found new blood relations. They are A blood group, B blood group, AB and O. The person who lives on other side of the globe is same with your blood group. It's called blood relative. Do you agree to this? AB group is human beings can receive the blood from any other group. Got it? The kit contains and the group anti-A sera 5 ml quantity, anti-B sera 5 ml, anti-RHD sera, porcelain. These all are the kit components. Blood kit should be ready in the hospitals. It is there. Some clinics, needle, Emmanuel in porcelain with white plate. Now there is a small experimental activity here. What are the materials required for it? Not included in the kit are cotton, 70% alcohol, toothpicks. Materials required. Not in the kit for this experiment. Alcohol and cotton toothpicks procedure. Take one porcelain plate, clean it and dry it. The plate must be very clean so that it does not interfere with the reaction. With a wax pencil, draw 3 circles on the plate to divide the surface into 3 parts. Draw 3 circles in each of them. Got it? So, AB is a human blood group which can receive the blood from any other group. Hence, it is called universal recipient. O group is universal donor. Who has O group can give it to anyone. Got it? The lab activity, when you find out the relationships, what we have to do, draw 3 circles. A group, A, B and RH should be written on third one. Okay, aim is to identify the blood group. What we have to identify here? The blood group of a person. So what we need? Blood group, identification kit, glass slide, wax pencil and disposable needle. With the wax pencil, draw 3 circles on a plate. Got it? To divide the surface into 3 parts and draw 3 circles. Put 1 drop of anti-serum at room temperature near the edge, but within each of the circle as shown in figure 14. Choose a finger, usually left ring finger. Clean this fingertip with alcohol like in a cotton ball and let it dry. Keep the cotton ball nearby as it is needed again. Dangle the hand down to increase the amount of blood in the fingers. Press on the button of the fingertip. Okay, when you have put 1 drop in 3 slides of blood corresponding anti-serum. Okay, with your thumb of the same hand, press it if the blood is not able to help hold blood in the fingertip and quickly prick it. Toothpick every time. The wax pencil circle will help to keep the sample isolated. Now what do you see? RBC's agglutination. They will appear grainy clumps, something like grains, small. See in the diagram of red blood cells, RBC suspended in a clear solution. RH is slower to agglutinate, so do not give up too soon. It will agglutinate slowly. So let it be. Determine the blood type group. With this experiment, we find out they are striated muscles, non-striated muscles, and cardiac muscle. You can move some muscles by our conscious effort. For example, the muscles present in inner limb move according to our wish and will. Such muscles are called voluntary muscles. We move it. Okay, these muscles are skeletal muscles and mostly attached to the bones and help in the body movement. Right? Whereas other muscles, which we are not responsible, they move without our control in our intestine, heart, small amounts of muscle tissue which are present in blood vessels. They help is increasing or decreasing the diameter of blood vessels, and thus the blood flows. Heart is made of only one type of muscle cells, and they help in pumping the blood. Got it? As a result, two types of cells are there, right? Voluntary muscle cells and non-voluntary, which are inside the intestine blood. We cannot control it, we cannot move it. Voluntary muscles in our hands and legs, skeletal system and all. Got it? Striated muscles diagram is there, non-striated muscles and cardiac muscles. Cardiac muscles are of, cardiac means of which the muscles which go to the heart. Okay, the bones which we move, the skeletal muscles, are light and dark band are called striations. As a result, they are called striated muscles. First one, which are present in our hands and legs, skeleton systems, striated muscles. Non-striated muscles, which goes inside the intestine, which we cannot control. Several internal organs have non-striated muscles. See the diagram? A little deviation is there, isn't it? Because of it goes in intestine, in our heart and all internal organs. Got it? Now make a tabular column like this. You will understand it better. The movement of food in the alimentary canal and the contraction and relaxation of blood vessels are involuntary. We cannot control it to go slowly or fast, isn't it? We cannot really start them or stop them simply by wanting to do so. Smooth muscles or involuntary muscles control such movement. They are also found in the iris of eyes, in uterus and its bronchi of lungs. Got it? Involuntary, unstriated. These cells, they are found in iris of eyes, in uterus, in bronchi of lungs. Underline it please and learn it. Smooth muscles or involuntary muscles. These cells are long with pointed ends. How would you keep water is, how would you know that water is hot or cold? If your leg on a sharp edged stone while walking, how will you feel if you fall down? The feeling in this situation is because of specialized mechanism in our body. It works like electric current passing through the wires. Brain, spinal cord and nerves play an active role in this mechanism. Is it clear? Now we see that the muscles present in the blood, again I am telling, are responsible for pumping of blood. These cells are branched and have nuclei. Cells are joined to each other at their ends. All the muscle cells in the cardiac muscles have striations. So they resemble striated in its structure, and it is involuntary. In the heart, in the intestine, in your uterus, lungs and all, they are involuntary. Means we cannot control it. They move there according to their timing and function they do properly. Got it? Brain, spinal cord, nerves have involuntary muscles. Got it? Now next activity 6. Collect a slide of nerve cells from the slide box. Now see that in the microscope. What do we see children in nerve cell? Nervous system is also important even in our body, isn't it? Nerve cells are the only cells in the body which do not have the ability to regeneration. They are very specialized cells. No two neurons or nerve cells in the nervous system have same appearance. Cells of nervous system are slightly specialized for transmitting the stimulus rapidly from one place to another within the body. We can identify 3 distinct parts. In the nerve cell there are 3 parts. Cell body or cyton, axon, second one, third one, dendrite. 3 parts are in this diagram. Dendrite first one, nerve cell, draw it properly, it can come for 4 marks, it's important question. Axon, and soma, revier nodes, myelin sheath, nerve terminals. Cell body or cyton has large nucleus and cytoplasm. The cytoplasm contains granular structure called Nissls granules. They are some projections arising from cell body. These are called dendrite. Got it, children? See the diagram? They are sharp, branched and more in number. One projection of cyton is somewhat longer than remaining projections, and this is called axon. The curved one is axon. Some nerve cells have axon covered with sheath-like structure. This axon covering sheath is called myelin sheath. Gas or nodes present at regular intervals on myelin sheath are called Ranvier nodes. Is it clear? 3 parts: cyton, axon, dendrite. Okay. Gas or nodes present at regular intervals on myelin sheath are called Ranvier nodes. See in the labelings. Draw it properly, color it, label it and send me today itself. Axon of nerve cell is connected with dendrites of a nearby nerve cell to form a web-like structure throughout the body. Is it clear? See the homework. What happens if the platelets are not present in the blood? Show the difference between 3 types of muscle fibers, children, with examples. Write the procedure to identify your blood group with the help of the kit. Collect the old blood reports of your friends, relatives and prepare a project report on the contents of blood. Draw a neatly labeled diagram of neuron and explain it. Thank you. Understood? Draw neatly and send by 5. Homeworks by 5. Thank you. See you in next class. Assalamu alaikum.